Good afternoon, everyone. On behalf of Amrita Mind Brain Center, I wish you all a convivial welcome to the afternoon session of the annual conference of Cognitive Sciences ACCS8 by Day 3. I hope you all had your break. So now we are again ready for our further sessions. The spirit of the event must go on, right? I hope all of us are as excited as we were in the morning. We are going to have a merry and enjoyable time together, like we had in the morning session. The most important thing when you're a student is that you should be able to interact with the prominent people in your study field. You should get a chance to listen to their valuable talks, their work, and their research. You should know what's going on currently in the research areas. I'm considering myself so lucky that I'm sure you are also thinking the same. As from the last 2 days, we are having this wonderful opportunity to listen to knowledgeable talks and sessions of such an amazing people. Further also, we'll be having 18 poster presentation, the Fireside Chat session, hosted by Professor Nitin Nagaraj from NIS Bangalore and Professor Vicky Baths from BITS Pilani, Goa, where we'll get to know their amazing work as well as current research and trends in the field of neuroscience and cognitive sciences. Followed by valedictory function, and lastly, the general body meeting, which will be open for all registered speakers and audience, so all can participate. So without wasting much further time, I would like to invite Ms. Arathy and Ms. Hemlatha from Amrita Mind Brain Center for chairing the fourth and the last poster session of ACCS 8.
Thank you, Mahima. Good afternoon, everyone. Let's start today's poster presentation. There will be 18 exciting poster presentations in this session. So the first poster is the role of BMI and perceptual load in attention capture by food stimuli by Ms. Rajeshwari Muthukumaran from IIT Bombay.
We are here to discuss the role of BMI and perceptual load in attention capture by food stimuli. Past research on attention bias to food stimuli has yielded ambiguous results. While many studies have shown an association between attention bias to food stimuli with craving, obesity, weight gain, and overconsumption of high-calorie foods, few studies show a weak or lack of association between attention bias to food and food consumption. Generally, these studies use dot probe or Stroop tasks, whose attention bias indices are often scrutinized for their reliability. Moreover, none of the studies manipulated the levels of attention to examine attention capture by food stimuli. Therefore, a recent string of studies measured attention bias to food from a perceptual load network. Morris and colleagues reported that the interference of food and food-related intrusive thoughts were reduced under high load conditions. Lastly, research shows a need for incorporating state, psychosocial, and behavioral measures such as BMI, diet quality, and hunger. The current study has done just that. Here we aim to understand the distractor interference of low and high calorie food in low and high perceptual loads based on high and low BMI. In our study, load was manipulated in the letter search task. Here we can see example trials of high and low calorie stimulus slides. The food distractors were present only 25% of the time, and the 47 participant data was divided into high and low BMI groups. And the error rates were further analyzed using 2 by 3 by 2 mixed ANOVA. The main effect of load was present as expected, and interaction between distractor conditions and BMI groups was also present. Here we can see a nice trend in the low BMI groups where a higher error rate was found for the low calorie density food, and in the high BMI group, however, a higher error rate was found for the high caloric density food. It is important to note here that the study is ongoing, therefore I'm presenting the notable trends. Next up further, we will be discussing the three-way interaction. So it is much clearer here that in high load condition, the low BMI group was distracted more by food with low caloric density, whereas the high BMI group was distracted more by food with high caloric density. We also observed a similar trend in low load high BMI groups. The current trends are in line with studies that showed enhanced orientation, longer engagement, and poor maintenance of attention towards high calorie foods. And this also contradicts the findings of Morris and colleagues. A few possible reasons could be that A, they used only high calorie sweet food, and B, they did not account for the BMI. Overall, the role of capacity limitations of attention appears to be an interesting new framework to study attention-biased food more reliably, and it could shed light on the cost and maintenance of unhealthy food consumption in those with higher BMI. Thank you.
Thank you, Ms. Rajeshwari. The next presentation is the benefit of knowledge generation using hands by Dr. Sok-Min Kang from the University of Texas.
Hello, everyone. This study explored learning benefit of generating knowledge using hands. There are many studies that showed the benefit of gesture, but especially there is a learning benefit in doing gestures. By using gesture, you can visually represent the internal understanding that is being refined through the process of description. Also, speakers who incorporated gesture into their learning process improved their own performance on various tasks and learning topics. So when speakers use gestures that are elicited as a product of describing concepts to others, they go through an additional cognitive process in order to generate the hands movement, okay? So they not only visualize a given information when they're using hands, but also attempt to simulate how it works through their body or hands, externalizing their thoughts. So through this embodiment process, their body and knowledge in mind become more strongly interwoven. From the perspective of depth of processing, when a learner observes a speaker and imitates a speaker's gesture, then the gesturing is regarded as being active, since the learner is cognitively matching his hands movement to what he observed from the speaker. On the other hand, when learners gesture without referring to a speaker's gesture when encoding information, it is a constructive learning process because they create knowledge with hands. So there are little studies that explore the benefit of gesture when a learner is asked to generate gesture without relying on a given object. This study explored the benefit of reading a textbook with and without the aid of gestures. Based on the research cited above, it is reasonable to expect that gesturing an idea using one's hands leads to a greater learning gain than just reading the text without gesturing. 33 participants participated in the study, and then there were two groups, no gesture group and then gesture group. Both groups took the pretest, and the gesture group read first for 10 minutes and then read quietly plus gesturing. And no gesture group read first and then again read the passage for 10 minutes, second time. And then both group took the post-test and then drawing test. In the result, there's no knowledge test differences between two groups. However, the gesture group showed a significantly better performance than the reading group. Gesture group drew more students in gesture group showed a completed diagram of human circulatory system than reading group. So generating gesture helps learners have a richer knowledge representation and give more learning benefits. Thank you.
Thank you, Dr. Sok-Min. The next presenter is Mr. Amarnath from IIIT Hyderabad, and the topic is Occasion Noise is Moderated by Expertise, Insights from over 1 billion open source chess games.
Hello everyone. Our topic is Occasion Noise is Moderated by Expertise, and we present to you the insights we have gained from analyzing open source chess games. So we are making judgments all the time, and studies show that when presented with the same evidence, judgments may be different on day-to-day basis. For example, in the book Noise, A Flaw in Human Judgment by Nobel laureate Kahneman, he describes how whenever there is judgment, there is noise, and much more than we think. The authors provide a very crisp measurement and examples of error breaking them down into noise and bias. While bias has been dominating the headlines, the authors show why noise is typically much bigger problem and ignored in research. For example, the influence of noise can be gauged from studies in medical domain, where it was found that even radiologists, they disagree on the mammogram readings around 50% of the time. And cardiologists disagree on artery blockages in angiograms in more than 30% of the cases. And same goes with dermatologists. Psychiatrists only agree that a patient has depression between 4 to 15% of the time. And prescriptions are influenced by the time of the day. The noise is broken down into a level noise. For example, some judges are more severe than others. Occasion noise. If faced with the same judgment, judges will not judge it identically. And pattern noise. Each judge has different preference and views on each cases. So based on these findings in the book, to the best of our knowledge, the research finding on noise are based on small sample size conducted in the lab or based on results from questionnaires and lack ecological validity. So we have taken gains and then analyzed them. Particularly, for example, for understanding the occasion noise, we have analyzed the prior result really influences the quality of the game, a subsequent quality of the game between novice, amateur and experts. Here we have taken similar strength players, and this strength is measured by ENO rating. And what we have found was that when we conducted the two-way NOI, it revealed that there was a statistical interaction between effects of expertise and game results. So between expertise, the prior result also influences them. So that is our finding. Though the impact of prior result on current task has been empirically found in simple tasks, there does not exist an exhaustive and ecologically valid research in complex tasks like chess. We plan to extend the sample size to much finer stratification of player strength. Thank you very much.
Thank you, Mr. Amarnath. The next poster is Visual Memory Enhancements Near the Hands by Ms. Lakshmi P. from University of Hyderabad.
Hello everyone, myself Lakshmi and I am from University of Hyderabad. So today I will be presenting on the topic Visual Memory Enhancements in the peri-hand space. So while hearing this title, you might have a lot of questions that is coming to your mind, like is visual memory enhanced near the hands? Can we make use of our hands in order to improve our visual memory and all of that? So I guess this presentation will give you answers at the end of this presentation. So let us move on. So past studies have shown how body and bodily spaces influence visual spatial processing of information from the environment. So the hand proximity effect has been widely investigated in the past decade, and two major accounts have been formulated for its underlying mechanism. So one is attentional prioritization account and the other is modulated visual pathway account. So attentional prioritization account basically clearly predicts that an enhancement for all visual tasks will be happening in the peri-hand space. And the modulated visual pathway account was formulated by Ghostley, and Ghostley proposes that placing an object near an action-relevant effector such as the hand would impose a bias processing in the favor of magnocellular pathway. And this is known as MDP account. So most of the studies that examine the peri-hand space effect have traditionally focused more on the aspects of the effects of hand position on attentional and perceptual processing. And in regard to the aspect of memory and some other higher cognitive processes such as emotions, so in this aspects, the studies is really very few. So only a very few studies have examined the effects of hand position on visual memory. So that's why we have chosen this as a main topic. So the goal of the present study is to carefully examine the possible enhancements in the visual short-term memory for the stimuli presented in the peri-hand space. So now let us look on to the methodology of this study and the experimental stimuli that is being used. So the main task that we performed was change detection task. So which is very simple, and it basically is a kind of exposing the participants to a set of things, and there will be a retention period, and right after that again this set of things will be presented or exposed to them, and they will be asked to detect whether there is any changes in this second trial. So that is change detection task in brief. So here in this change detection task, there was mainly two hand conditions that we have provided. One is near hand condition and the other is far hand condition. So the stimuli type that we used was arrays. So there was a memory array followed by a test array with a retention period or retention interval in between. So the array consisted of 4 lines. So the size of the lines was 3 centimeter in length and 0.7 centimeter in width. And there was a visual angle of 2.93 into 2.35 visual angle. And the colors that we have chosen for these lines were blue, green, red, yellow, and orange. And there was change type, which was there was an orientation change happening, followed by color change, and we induced both this orientation and color change in the trials separately. So the array of bars were presented on a LCD monitor with a screen resolution of 1920 into 1080 with a screen refresh rate of 60 Hertz. And the participants were asked to sit in front of a laptop, and keeping their hands on the monitor was referred to as the near hand condition while performing the task or while responding. And when the participants were asked to keep their hands on their lap, and that was considered as a far hand condition while responding or while making a response. And the responses were made on the mouse by pressing one of the 2 buttons in each mouse. So I will, for a clear understanding of this methodology, I will show you the trial design. So here we go. So this is the trial structure that we have followed in the experiment. So there was a fixation in the screen, and this whole experiment was performed in a dimly lit room. And the fixation was appeared for about 500 milliseconds, and that was a cross mark at the middle of the screen, followed by a memory array which consisted of the 4 lines of varying colors, and that was 100 millisecond duration. And a 1000 millisecond blank screen, which was the retention period, was also presented, followed by a test array. And the test array might vary, either it will be an orientation change or it will be color change or a both, or there was also a no change condition that was also 100 millisecond duration. So in prior to the experiment, the participants were not informed about what all changes will be happening and all of that. So that was ensured. So this is the trial structure. Now moving on to the results part. So a total of 15 participants were being a part of this experiment, where 8 males and 6 female participants were there in the experiment with a mean age of 22 years. So the task was performed among all the participants, and the data was being collected, and we analyzed the data mainly focusing on the accuracy. So the dependent measure of the interest of the study is sensitivity, that is being denoted as D prime. So that was used as an index of measurement of visual working memory. So the accuracy data was calculated as a function of change type, which is color, orientation, and both, and hand position, which is hand condition and non-hand condition. A2, hand position and near and far, into 3, that is change type. Repeated measures and over was performed on the D prime values obtained. While doing the analysis of D prime, there was 4 conditions that is being considered. We have calculated the hits, false alarm, and correct projection, and the misses. So all these were calculated in order to figure out the D prime value. And as the results suggest that there was a significant main effect for the hand position, and the numbers is denoted as given below. And also the findings shows that the D prime sensitivity to the changes type were higher for hand position for hand condition compared to the no hand condition, implying relatively better retention of information presented in the hand space compared to the no hand space. So this is a graphical representation or illustration of the results. That is, in the x-axis we have the hand position, this is hand condition, that is hands near condition, and hands far condition, that is no hand condition. And in the y-axis we plotted the D prime values. So we could clearly see, like we could clearly see the orientation and color and both the conditions, how it is varying. So in the hand condition, the hand position, in the hand condition, the orientation and all of these values were more, when in no hand condition it was very less. So we could clearly see that there was a positive effect of hand condition when compared to no hand condition for this stimulus. So from this small experiment, the results obtained clearly suggest a strong main effect for hand position. The D prime scores indicates a higher sensitivity for stimuli present in the peri-hand space than the far-hand space. The current experiments findings are in line with the MVP hypothesis that we have discussed in the introduction part, which suggests that hand proximity effect is enhanced for orientation information than color information. So here we could clearly see in the graph, for orientation information, it is more than compared to the color and both condition. So yeah, the results obtained in the study are also in line with the Kelly and Brockmore study, which was conducted on 2014, except for the effect of color information. So Kelly and Brockmore has suggested that in near hand condition, orientation information and color information, orientation information will be enhanced more, whereas the color information will be enhanced more in far hand condition. But here in this experiment, in our experiment, we have clearly got a result that orientation information is being enhanced more in near hand condition, similarly to the color condition also. So also this study included a third feature, which is different from Kelly and Brockmore's and previous studies. So we have introduced a new feature, which is the combination of both the color and orientation information in the trial type, named as both. So the statistical results suggest that there was no significance for both the trial types in peri-hand space, and the reason for that is still unclear. We might need further studies to investigate why there was no any significance or there was no significant effect for this feature. So in order to conclude, the present study indicates a significant main effect for hand position in visual memory for stimuli present in the peri-hand space. So the D prime sensitivity used as visual memory index in this study. So the D prime sensitivity to change types were higher for near hand or hand condition compared to the far hand condition, even though the experiment design of the study have many advantages over the previous studies, such as there was no any across comparisons because all the trial types were clearly separated in order to find a clear understanding of how orientation is influencing the hand condition, how color is influencing the hand condition and all of that. But there are some limitations also for this study, because change detection task in itself is so much demanding, demanding a lot of complexities involved, so which might affect the memory capacity or the memory retention capacity of the participants and all of that. So there are a lot of limitations also. But however, our approach, we believe that our approach allows us to ask additional theoretical questions which have future scopes as well, such as whether hand positions affect one's ability to bind the visual features. If yes, what is the neural mechanism for this binding process, which helps to create a unified representation of objects in the visual field? So that is a potential question. And the other is, what will be the effect of hand position on visual memory? The working memory load is increased. So if you are increasing the amount of stimuli that is being presented to the participants, will that have an effect on the orientation? Will that have an effect on memorizing this stimulus? And will there be an influence of hand position effect on visual long-term memory for various visual features? So as an overall, we could clearly state that clearly state that the hand position is having an influence on visual memory and enhancement. So that's the main finding of the study.
Thank you, Ms. Lakshmi. The next presentation is a study on Parkinson's disease under the context of reinforcement learning by Ms. Sasidharakurup from Amrita School of Engineering.
Good afternoon. Myself Srija SR. I'm a research scholar at Amrita School of Engineering, Amrita. Today I'm going to give a poster presentation on my work, a study on Parkinson's disease under the context of reinforcement learning. The work is motivated by the objective slide to study the dynamic behavior of Parkinson's disease using a fully artificial reinforcement learning model, to introduce the role of overactive acetylcholine in Parkinson's tremor, and then to visualize the Parkinson's tremor in Unity platform with the help of virtual robot, and finally to propose a generalized agent environment method to study the pathway related problem in brain. The work is all about integration of artificial intelligence and neuroscience. So to understand the concept of reinforcement learning in brain, we are focusing on a part of the brain basal ganglia and the neurological condition associated with the lesion of basal ganglia, Parkinson's disease, which occurs due to depletion of dopaminergic neurons. As we know, in artificial intelligence, unlike the supervised and unsupervised learning approach, reinforcement learning is an agent-based learning where the agent interacts with the environment and moves to the next stage based on an action that maximizes the expected future reward. The entire work is divided into 3 major modules. The first one is about mathematical formulations of factors affecting PD. Second, about RL model implementation. And third, about demonstration of tremor with the help of virtual robot. In module 1, we'll be considering some factors affecting depletion of dopamine like acetylcholine imbalance, oxidative stress, formation of Lewy bodies, gene mutation, etc. So primarily we'll be focusing on the imbalance of acetylcholine because a decline in dopamine neurons in brain may lead to excessive acetylcholine that may lead to Parkinson's disease tremor. So once we get the mathematical formulations, we'll be moving to the next module, module 2, which is about the implementation of RL model, where we express the problem as Markov decision process. Here, we'll be proposing a generalized method to create an environment, introducing these factors or mathematical equations as environment variables for the agent to learn, so that we can use the same approach to study other pathway problems in brain. Parts of the basal ganglia here are considered states, and neurons in brain are considered as agents, and action is selecting direct or indirect pathway. Thus, by mapping the mathematical formulations, that is factors affecting depletion of dopamine with state action pair, we expect the agent to learn these factors and take an indirect pathway resulting in tremor. Finally, the demonstration of the tremor and visualization of value iteration can be done with the help of a virtual robot implemented in Unity platform. These are some of the references. Thank you.
Thank you, Ms. Srija. The next presenter is Dr. Vishwa from Berlin Institute of Technology, and the topic is Deep Learning Framework for Casual Connectivity-Based Epileptic Neuromarkers from Multi-Channel EEG.
Our work is to develop deep learning framework for causal connectivity-based epileptic neuromarkers from multi-channel EEG. Objectives are to predict an epileptic seizure onset using EEG, which translates into classification of an EEG pattern as preictal epileptiform, and secondly to prescribe neuromarkers of epilepsy. Preictal slowing and spiking of signals in the literature have been shown due to long-range time-varying correlations, and hence phase locking value has been chosen as the feature describer, which measures the instantaneous coherence of 2 signals. The method involved was automized data pre-processing and data augmentation, visual and statistical significance of discriminative patterns of effective connectivity, and then the training and testing of the 3D CNN based binary classifier. The workflow shown here in this diagram has been described in the further slides. First of all, data was divided into pre-ictal, interictal, and ictal periods. Then noise removal by applying notch filter was and normalization was also performed. Data augmentation by cutting and splicing was performed to handle class imbalance. Based on the knowledge of default mode networks, 6 regions of interest were selected, and the mean phase locking value for all time points in each epoch was computed for different bands. A faster algorithm for computing the mean PLV was adopted from reference research, and data-driven thresholding was performed to filter out significant features. Here we can see distinguishing patterns of connectivity among certain locations can be seen in the interictal, pre-ictal, and ictal patterns of mean PLV values. And these have been actually used by the model to learn the distinguishing patterns and do classification. Statistically significant distinguishing patterns were found among certain electrode regions and in pre-ictal beta band, interictal gamma band, and pre-ictal beta band and interictal beta band. This contrasts with the traditional emphasis on coupling among the demons in gamma band only. The model's accuracy was high, and low false prediction rate was achieved when tested with single subject data, but with multi-subject data, some reduction in validation accuracy was seen, and this takes us to further work. Data from MIT Physio Data was used.
Thank you, Dr. Vishwa. So the next presentation is by Arathy Rajendran from Amrita Mind Brain Center. The presentation is on cerebellum model circuitry explaining knockout behavior of Purkinje neuron during movement disorder.
Good afternoon all, I am Arathy Rajendran from Amrita Mind Brain Center. Today I am going to talk about cerebellum model circuitry explaining knockout behavior of Purkinje neuron during movement disorder. Autism spectrum disorder is a collection of neurodevelopmental disease which is the result of the combined action of both genetic and environmental factors. The main symptoms of ASD include delayed communication skills, impaired social behavior, hyperactivity, and unusual motor emotional reactions. According to the World Health Organization, 1 in 160 children has autism. Hence, autism research will be helpful for autistic patients as well as for the early diagnosis to find the cause of other neurodevelopmental disorders. Several studies in autistic patients have shown that the cerebellum has an important role in the development of new motor-related symptoms from structural and functional neuronal abnormalities due to ASD. Previous reports have indicated that the cerebellar Purkinje neurons were reduced around 95% in most of the cases of ASD when compared to the control. The genetic level manipulation allows researchers to study the signaling pathways in all types of cells, including neurons. Hence, the Peter knockout study is important for discovering the role of cerebellum in ASD. Peton is a phosphate and tensine homologue that has a role in maintenance of genomic stability, cell migration, survival, and proliferation. In this study, the electrophysiological characteristics of Purkinje neurons based on the structural and functional changes that occur after Peton knockout have been reconstructed to predict the neuronal activity associated with Purkinje cells in the cerebellum network. Here, we have used a spiking neuron model of the cerebellum network due to its simplicity and computational efficiency to simulate large-scale neurons. The neurons of the cerebellum were placed in a virtual 3D space to mimic the cerebellum layers, including molecular, Purkinje, and granular layers. All the cerebellum neurons were connected based on the convergence-divergence ratio, which was obtained from experimental data. The simulation was performed for 3250 milliseconds and divided into 5 stages of ASD as seen in the Peton knockout mice. In the early onset, the spontaneous firing of the PC became reduced and the firing started to be abnormal. The interspike interval becomes high as it progresses till the 9 months. The result showed that from the 9 months, the Purkinje cell neuron was reduced and the number of stellate and basket became reduced to exhibit the reduced thickness of molecular layer. During the 12th month of disease condition, the Purkinje cell number became too low to represent cell death. This study showed that the mutated PC means Purkinje cell also modulates the deep cerebellar nuclei in abnormal ways. So the main aim of this study was to build a spiking neuron network of the cerebellum, and this model is used in the study, helped us to understand the electrophysiological characteristics of Purkinje cell in the cerebellum during AST conditions by mimicking the knockout behavior in Purkinje cell. This would result in improving clinical diagnosis, therapeutic interventions, and strategies. Thank you.
Thank you, Ms. Arathy. That was a very interesting study about knockout modeling autism. If there are any questions, they can be asked after the session. The links will be available on the ACCS8 website under the program schedule. Please visit www.amra.edu/ACCS8. So the next presentation is to be on stability of sensory motor networks curves, the dynamic repertoire of resting stage across age by Ms. Nisha Chetana Shastri from NBRC Manesar.
A very good day to all. I am Nisha Shastri. In this poster session, I will be presenting our work where we investigate temporal stability of DFC patterns during rest and task across lifespan aging. We also investigate the stability of resting state networks across aging as well. Aging is typically associated with substantial structural and functional changes in the brain. Several studies indicate that stable representation of neural activity and the corresponding stability of FC patterns is very crucial for survival. All the previous studies have explored the association between DFC and age. How the stability of functional architecture modifies across lifespan aging remains an open question. In this study, we address this by investigating the modifications in temporal stability of resting state and task with a novel unsupervised data-driven method. In this study, we employ a data set of 645 participants from CAMCAN consortium consisting of resting state, passive movie watching task, and sensory motor task data. We divide the data set into 3 cohorts, young, middle, and older adults. Coming to our methodology, we calculate dynamic functional connectivity using bold-face coherence, and we capture the higher dimensional dominant DFC patterns in lower dimensional subspaces by employing a dimensional reduction technique, and subsequently calculate the temporal stability between these lower dimensional dominant DFC subspaces using 2 metrics, that is 1 angular distance and 2 Mahalanobis distance. We calculate the angular distance between dominant DFC subspaces of rest, movie, and sensory motor task. The time cross time temporal stability metric, each entry in this matrix, is the principal angle between the dominant DFC subspaces. Smaller the principal angle, similar the DFC subspaces, hence they are more stable. What we find in our analysis is, we find a global widespread temporal stability patterns of shorter duration in resting state, a local abated but longer duration temporal stability patterns in the tasks in all young, middle, and older adults. We verify these results by calculating Mahalanobis distance between dominant DFC subspaces. Each entry in this Mahalanobis distance metric is each entry in this time cross time temporal stability matrix is the Mahal distance between dominant DFC subspaces. Smaller the Mahalanobis distance, similar the DFC subspaces, hence they are more stable. We find that our results with Mahalanobis distance metric validate our results with angular distance metric. Next, we quantify the temporal stability patterns as seen in our temporal stability matrices using entropy. Overall, as illustrated by figure 3a, we find that temporal stability of FC in tasks was higher than the resting state. Across lifespan aging, what we find is, in resting state especially, we find the characteristic U-shaped trajectory, that is peak entropy in middle adults followed by young and old adults. Whereas in task, we find peak entropy in older adults followed by middle and young adults. Next, we proceed to investigate lifespan aging specific changes in stability of resting state networks. We find a significant decrease in temporal stability of central executive network and visual network, whereas we find a significant increase in temporal stability of sensory motor network. We also find a significant increase in temporal stability of whole brain resting state network. Moreover, we find that the stability of whole brain resting state was primarily influenced by stability of sensory motor network. To conclude with our study, what we were able to establish the following. One, we introduced a novel unsupervised data-driven method to establish temporal stability of DFC patterns. Two, we found a global widespread shorter duration temporal stability patterns in resting state. Three, the stability was highest in sensory motor task, followed by movie watching and resting state. We found a significant decrease in stability of central executive network and visual network, but a significant increase in temporal stability of sensory motor network. More importantly, we also found that the stability dynamics of whole brain resting state closely follows the stability dynamics of sensory motor network across lifespan aging. Thank you.
Thank you, Ms. Nisha, for the presentation. The questions can be asked at the end of the session. We have our next presentation by Mr. Sridev Radhakrishnan from Amrita Mind Brain Center. The presentation is on mathematical modeling, activity dependent vascular changes in cerebellar granular layer. Let's move to the recorded video.
Good afternoon all. I am Sridev, PhD student at Amrita Mind Brain Center. As part of the 8th Annual Conference of Cognitive Science, I am presenting this work entitled Modeling Neural Activity Dependent Vascular Changes in Cerebellar Granular Layer. Human brain, being the most metabolically active part of the body, requires continuous supply of nutrients and oxygen through local blood vessels. The reconstruction of neural activity dependent local blood vessel diameter changes in brain has not been explored much due to complex interaction between parameters and heterogeneous behavior. Hence, in this study, we use mathematical understanding to connect models at several levels, especially from underlying neuronal population activity to the blood flow changes, and from the nitric oxide level towards neurovascular coupling. And also, the influence of production of nitric oxide in arterial radius changes were also explored to understand the relationship between changes in blood flow and volume during neuronal activation. This figure explains the feedforward arterial blood vessel changes wherein synaptically released glutamate-induced NMDA receptors activation leads to calcium influx, causing the release of nitric oxide from neuronal nitric oxide synthase, which activates a smooth muscle guanylate cyclase. This, in turn, generates cyclic CGMP to dilate the arteriolar blood vessels. Detailed multi-compartmental biophysical granular cell model with 52 active compartments were used to model the changes in arterial radius, and the modeling was done using neuron simulation platform. Arterial radius profile changes were modeled by varying synaptic inputs. For this study, we provided 2 synaptic inputs and recorded the corresponding voltage changes and nitric oxide profile changes, and also modeled the radius changes in correspondence with the changes in neural activity and nitric oxide profile. To record the changes in radius profile with respect to the varied input, we provided 4 synaptic inputs, and an increase in arterial radius from 0.8 to 13.15 micrometer was observed by changing the initial radius of 0.6 to 1.5 micrometer by providing 4 synaptic inputs. Step input of 40 millivolt from 400 to 600 seconds was provided as a stimulus to validate the result, and the corresponding changes in arterial radius was matched with the experimental data from Dormans et al. 2016. To conclude, mathematical modeling helps connect models at several levels, especially from underlying neuronal population activity to the blood flow changes. Granular cell activity causes a robust vasodilation in the nearby capillaries via NMDA-mediated neuronal nitric oxide synthase signaling pathway. Through this modeling study, the influence of nitric oxide in neurovascular coupling was explored to understand the relationship between changes in blood flow and volume during neuronal activation. Although several aspects need to be included, such as arterial dimension, extension of vascularity, diffusion of nitric oxide in networks. This can easily be extended to study various behaviors relating animal experiments to human neuroscience and may serve as a step for mathematical modeling in physiology. Thank you all.
Thank you, Mr. Sridhar, for the presentation. The questions can be asked at the end of this session. The links are available on the website www.amra.edu/ACCS8 under the program schedule next to the titles of every poster. So let's go to the next presentation on computational modeling of neurovascular coupling and fMRI BOLD correlates the neural circuits using BOLD SIM by Mr. Chaitanya Nuttaki from Amrita Mind Brain Center.
Good afternoon, everyone. I am Chaitanya Nuttaki, PhD student at Amrita Mind Brain Center. As part of 8th Annual Conference of Cognitive Sciences, here I am going to present a part of my work on computational modeling of neurovascular coupling and fMRI BOLD correlates of neural circuits using BOLD SIM. In this study, we have proposed a hemodynamic model as a tool called BOLD SIM that can analyze the concentration profiles of nitric oxide generated from single or multiple sources of neurons and the correlation between the neural activity and blood flow changes in terms of fMRI BOLD responses. BOLD SIM was designed as an easy to use tool for computing biophysical and physiological BOLD responses of single neuron and a population of neurons. BOLD SIM tool interface was created using neurons built-in graphical components which allows user to view neural activity and computed BOLD CBV changes along with morphology view window. As the field of fMRI has evolved, several hemodynamic models have been developed. In this study, the changes in neural activity to biophysical BOLD responses was modeled using a combination of balloon and Windkessel model with a set of ordinary differential equations. The developed model was better able to capture the flow and volume temporal changes in terms of arterial compliance. Also, nitric oxide mediated pathway that controls the vessel diameter changes with respect to neural activity was modeled using first order nonlinear differential equations. And the platform used in this study is Neuron. The models which we used to test the BOLD SIM tool was detailed granule cell model, morphologically simplified detailed granule cell model, layer 3 and layer 5 pyramidal neurons. The BOLD responses can vary from voxel to voxel and subject to subject, and these variations were accommodated in the current model. Using a computational model of cerebellar granule neuron, BOLD SIM was evaluated with different firing frequencies. In the figure, A shows the neural activity, B and C shows the BOLD and cerebral blood volume changes with respect to the provided neural activity. Since the blood flow and synaptic activity were interrelated, the model flow to volume and BOLD responses in the cerebellar cortex expressed a seemingly linear relationship to the stimulus duration. The temporal mismatch between the blood flow and the blood volume changes as delayed compliance was incorporated into the model with an exponential function estimating its temporal evolution using volume and blood flow measurements. Nitric oxide concentration and blood flow changes with respect to the neural activity was modeled using BOLD SIM. The role played by nitric oxide in neurovascular coupling at mossy fiber granule cell relay was investigated by varying different inputs. Multi-compartment granule cell providing 4 mossy fiber synaptic input with a total simulation of 1000 milliseconds was used to generate the responses. The initial dip and rise of nitric oxide concentration, BOLD and CBV changes was according to the stimulus and post-stimulus model. Simulations indicated that increase in the nitric oxide induced neural activity leads to increase in the blood flow, volume and BOLD concentrations that helps in more dilation. In the modeling, an increase in the CBF leads to increase in amplitude changes of BOLD CBV responses with respect to the corresponding changes in the local neural activity. Neural activity dependent physiological parameters and the vasoactive agents that helps in vasodilation and constriction. As a future perspective of this work, with the emergence of this bottom-up reconstruction of connecting neural activity to fMRI BOLD and CBV responses, this BOLD SIM, the developed BOLD SIM still need more detailed experimental validations of the neurovascular coupling components and that surface in physiological and biophysical bold responses. Thank you.
Thank you, Mr. Chaitanya. We have our next presentation by Ms. Nijin Nisar on implementing web-based bioinformatics and biosignal analysis virtual laboratories for neuroscience education in universities. Let's move to the recorded video.
Good afternoon all. I am Nijin and on behalf of my colleague Mr. Danish Kumar, here to present the poster on implementing web-based bioinformatics and biosignal analysis virtual laboratories for neuroscience education in universities. Rapid growth of information technologies in India propelled the implementation of digital e-learning resources in science and engineering educations. Government of India has been taking initiatives for developing diverse e-learning resources such as virtual laboratories to provide experimental skills for science experiments and for analyzing scientific data. These web-enabled experimentations were developed and deployed for enhancing digital education and immersive learning experiences. Due to crisis of COVID-19 pandemic, education institutes were forced to adapt and implement technology-based e-learning platforms in their curricula aimed to provide a sustainable education. This content-rich learning platform includes theory, experimental setup, equipment training, interactive animations, simulations, and assessment questions as examination component to reproduce conventional laboratory experience with relevant pedagogical features. With the focus of delivering realistic virtual environment for learners to get a hands-on experience on performing biosignal processing and data science, we developed and deployed two new virtual laboratories: Biosignal Processing and Analysis Lab and Bioinformatics and Data Science in Biotechnology. The main focus of EEG Simulator Lab is on processing EEG signals and events that is aimed to help students to analyze and classify event-driven variations with specific tasks such as motor tasks, visual perception, working memory, and cognitive aspects of yoga and meditation for understanding neural correlates underlying specific cognitive activities. Bioinformatics and Data Science in Biotechnology Virtual Lab focuses on the basic practical techniques in bioinformatics and bioscience data processing using basic research applicable educational methods. The lab is designed to provide practical training in bioinformatics methods, including retrieving public sequence databases, use of different computational tools to find sequences, analysis of protein and amino acid sequences without the need of any other software packages. Successful development and implementation of such lab will diversify interdisciplinary curricula in the neuroscience and data science to understand functional neural underpinnings of human behavior. Thank you.
Thanks a lot, Ms. Nijin, for introducing us to virtual laboratories. Of course, it would be really helpful for students, especially during this pandemic. So the questions can be asked at the end of the session. Next, we have the presentation on learner perception and preferences of using virtual laboratories in STEM education pre and post COVID-19 pandemic by Ms. Rakhi Rathamani from Amrita Mind Brain Center.
Warm good afternoon all. I am Rakhi Rathamani, hereby presenting my poster titled Learner Perception and Preferences of Using Virtual Laboratories in STEM Education Pre and Post COVID-19 Pandemic. It has been noted that the crisis of the COVID-19 pandemic forced the education institutes to adapt and implement technology-based e-learning platform towards a strategic plan to bring evolution and change within higher education system. While theoretical concept could be covered efficiently through online mode, conducting laboratory courses at university level still posed challenges in the context of sustainable development goals, the potential factor to meet quality education. According to Cognitive Framework for Learning, the use of innovative multimedia technologies with interaction elements were preferred for improving learner perception and to make learning effective and productive. Virtual laboratories are massive open online courses providing content-rich learning platform including theoretical aspects, experimental setup, equipment training, interactive animation and simulations and assessment questions to reproduce conventional laboratory experiences with relevant pedagogical features. The present study focuses on analyzing user behavior and acquisition on using virtual lab platform pre-COVID-19 days with blended learning approach and post-COVID-19 times with remote learning approach. Using term and OER based behavior analysis, the study included university students N is equal to 1059 in blended learning approach pre-COVID days and online users as study participants during COVID days. The initial study showed transformation of conventional education with minimum instructor dependency using blended approach before COVID-19 days. Usage analysis indicated implementation of virtual labs as an interactive textbook for understanding theoretical and experimental concept and user-centric approach in conceptual learning with minimum instructor support. Comparison on usage time, user access time bins indicated user intention as assimilators. They can complete exercise without the instructor support and need an audio or video presentation for abstract conceptualization and reflective observation. Online usage analytics during the COVID-19 pandemic time indicated rapid increase in the number of registered online users during the lockdown phase. Adoption of virtual labs in learning and teaching scenarios were studied using factors such as number of users, number of sections, page views, unique views and bounce rate. Studies indicated a logistic S-shaped curve for virtual laboratories adoption among students-teacher community with an early slow phase, a rapid middle phase with widespread and a slow third phase with an incomplete penetration in the end. This work is funded by Ministry of Education, Government of India. I would like to acknowledge my colleagues, Mr. Danish Kumar and Ms. Nijin Nisar for helping me in data collection and analysis. Thank you.
Thank you, Rakhi, for the presentation. Our next presentation is on computational drug repurposing approach to identify potential DPP-4 inhibitors to develop novel anti-Parkinson therapy by Mr. Parag Vashne from GLA University.
Good afternoon, everyone. Myself Parag Vashne from GLA University, Mathura, and my title of presentation is Computational Drug Repurposing Approach to Identify Potential DPP-4 Inhibitors to Develop Novel Anti-Parkinson Therapy. In my paper, I found some drug molecules that has binding affinity with DPP-4 enzyme to inhibit them and maybe for candidate to treat Parkinson diseases. We know that drug has multi-purpose role to treat Parkinson, to treat various diseases. For example, aspirin has analgesic, antipyretic, anti-inflammatory, antiplatelet effect. Through repurposing or docking process, we can explore our existing drug molecule to find new therapeutic indications. And it is a very cost-effective process and reduced drug development time because we can skip a preclinical trial because already have pharmacokinetic profile, safety, efficacy of already existing drugs. In docking, we find orientation of existing drug molecules so it can bind to the receptor by stabilizing of complex and minimum release of free binding energy. The binding energy should be between -5 to -15 kilocalorie per body so that it can make a reversible complex. And I work on DPP-4 enzyme inhibitor to treat PD diseases. And DPP-4 enzyme is responsible for the degradation of various incretins like GLP and GIP. When we inhibit that enzyme, the GLP and GIP level increase in our body and cause amelioration of Parkinson's diseases and dopamine level increase in our body. I have done docking for to find preferred orientation and binding energy of our drug molecules for to inhibit DPP-4 enzyme as a novel anti-Parkinson therapy. So firstly, I obtained 3D structure of DPP-4 enzyme with ligand through RCSP protein database bank type. After that, I separated this ligand enzyme by the software Chimera. After that, I prepare ligand and receptor and also done docking by making grid box center to the ligand by covering all the residues wrapped with the ligand. In Autodoc software, after that, we make docking parameter file and obtain all the results like binding energy and what are the interaction with their amino acids. After that, I validate all the parameters by overlay method and chemical resemblance method. After that, we taken a library containing 2890 FDA-approved drug molecules from the database and screened through TPP4 enzyme. And I found that many drug molecules in which mesligine has minimum binding energy minus 9.34. Mesligine has minus 9.43 binding energy, and it may be a preferred candidate for to treat Parkinson's disease. Finally, I conclude my paper that the drug development takes too much time, like 15, 20 years and $2 to $3 billion. But we can reduce cost or development time by in silico docking techniques. Thank you for to listen my presentation.
Thank you, Mr. Para. That was an interesting study. The questions can be asked at the end of the session using the website link available on www.amrita.edu/ACCS8. So the next presentation is on temporal structure on heart-brain interactions across adult lifespan by Ms. Kirti Saluja from NBRC, Manesa.
Good afternoon, everyone. Thanks for dropping by my poster. So brain said to the heart, we need to talk. So while they are having their little conversation, let's just wonder a little bit about how these two organs, which have their intrinsic oscillatory properties, interact with each other. And does age affect this interaction or not? Because there are a lot of literature which suggests there's cardiac dysfunction and there's a lot of changes which happens in the brain rhythms across age. So this project is an attempt to gain more integrative view on the heart-brain dynamics. So we were interested to study the following questions using a lifespan data set from Cambridge Center for Aging and Neuroscience. So we used resting state, MEG, and ECG recording. So the first question we asked is, how does the heart evoke potential, which actually represents the cortical processing of the heartbeat, changes across age? Further, we were interested into studying how does the features of the spontaneous brain activity time locked to the R peaks changes across age. So for that, we employed this very effective technique of Syncograms. So basically, it's a technique which helps us to understand phase synchronization between a point process, in our case, it's the heartbeat, and a continuous signal, that is the brain rhythms. So, and then we used Hilbert transform to extract out the phases, time locked to the R peaks, and saw its trend across age. So the HEP analysis revealed there's age-related differences in the cortical amplitude time locked to the R peak. Here we can see that the cortical amplitude at the R peak decreases across age, while the cortical amplitude between 240 to 320 milliseconds, which actually represents the interoceptive awareness, it increases across age. Further, we saw that the phase values of the brain oscillations time locked to the R peak of the ECG time series showed a positive trend across age in theta and alpha brain rhythms significantly, which actually we can speculate that there's a differential interaction happening between the brain rhythms and the cardiac signals. Hence, our results provide initial framework to explain how the cardiac signals are processed and represented at the cortical level. It can actually provide us a window to understand the heart-brain interaction and its role in shaping our cognition. Thanks a lot for your attention and time.
Thank you, Ms. Kirti. That was an interesting presentation connecting the heart and brain dynamics relating to age. So if there are any questions, it can be asked at the end of the session. Next, we have the presentation on multimodal and interpretable diagnostic cum therapeutic framework for epileptic seizure prediction by Dr. Shaurya Sriyam from IIT Delhi.
Hello, this is our poster presentation. We are trying to predict epileptic seizures. EG data is the most commonly used data for this purpose, and we'll be building a novel framework around this. So we basically wish to predict the starting of the pre-ictal phase as early as possible and as accurately as possible. This may turn out to be a huge lifesaver for epileptic patients. And we also wish to use fMRI data for this purpose because fMRI has very good spatial resolution, and this may help us locate the region where the epileptic spikes are getting initiated. And for this purpose, to work with EEG and fMRI data, we need to adopt a deep learning approach. And in deep learning approach, generally people explore convolutional and recurrent neural networks, but we wish to also explore spiking neural networks because spiking neural networks are relatively less explored, and they have been shown to be very useful in instances of complex recurring patterns of different types. But the one major drawback with deep learning approaches is that they are black box type of machine learning techniques, and therefore they are not preferred by neurologists and radiologists. Therefore, we wish to make it more interpretable, and there are frameworks like SHAP, Deep SHAP, Kernel SHAP, which have been created for this purpose, but they have not been used a lot in respect to EEG type of machine learning task. Therefore, we want to do this. And to summarize, to end this, we don't want to limit ourselves to diagnostic purposes. We also wish to use our framework for therapeutic framework. From this, what we mean is that most pharmaceutical companies today are trying different kind of drugs and invasive, non-invasive treatment techniques to help these mental health patients, and they try to they have formulated several quantitative metrics based on which they will track the improvement of the patient's condition. But there are not enough such micro level quantitative metrics, and we believe that by using gene expression data, we can augment the radiologists and neurologists with these micro level quantitative metrics, and this can be used for personalized healthcare objective, as well as it can be used to identify causally relevant biomarkers for epileptogenesis. But integrating these three types of data, EEG data, fMRI data, as well as genetic data, the biochemical level of gene expression, proteomic data, will be very challenging, but we believe this will provide a comprehensive diagnostic cum therapeutic framework for the purpose of improving the lives of epileptic patients. Thank you very much for attending our poster presentation.
Thank you, Dr. Shaudin Sriram, for the presentation. The next presentation is by Dr. R. Guru Ravi Shankar from IIIT on music emotion recognition via deep learning and comparison with human perception.
Hello all, welcome to the video presentation of our work, Music Emotion Recognition via Deep Learning and Comparison with Human Perception. Music plays an important part in our daily lives. Individuals seek varied emotional experiences via music. Identifying emotions from music helps in organization, retrieval, and recommendation of music to satisfy an individual's personal needs. So, music emotion recognition is an important task in the field of music information retrieval. However, MER has been limited to the usage of acoustic content, social tags, and metadata. Lyrics have been neglected in MER, even though they play a crucial role in eliciting emotions and also contributing musical reward. The recent advancements in natural language processing techniques, especially the transformer model, has helped improve MER models on English lyrics because it ensures longer dependencies. The number of studies on MER for Indian languages is less when compared to English, but none of them used transformer models for MER task. In this study, we evaluate the efficiency of multilingual transformer model on MER task in Hindi. We use the Bolly dataset, which comprises 1055 song lyrics from 1970 to 2017. Each song is annotated for emotion by 3 annotators solely based on lyrics without listening to the audio. We use a multilingual transformer model, XLM Roberta, which is pre-trained on 100 languages, including Hindi. We fine-tune the pre-trained model on the Bolly dataset. Hence, given the lyrics in Hindi, the model outputs whether the lyrics is positive or negative. We use the same train test split of 90-10 as used by the original authors to compare the performance of the model. We report a superior average accuracy of 80.12% compared to 75.49% by the SPM model. To understand the better performance of XLM Roberta and OSPM, we analyze the misclassified lyrics of the best XLM Roberta model in terms of F1 score and accuracy by using perceptual validation of two annotators. The annotators agreed with the predictions of 50% of the misclassified song, of which 2 of them are given. Also, few of the misclassified song lyrics had both positive and negative aspects, rendering ambiguity. In this study, we demonstrate the efficiency of using XLM Roberta model on MER task in Hindi. The observation from perception prediction task suggests that there would be some amount of disagreement with respect to the ascribed emotion of given lyrics between the annotators. Though the annotators were not made to listen to the audio of the songs, there would be some familiar songs whose audio features may confound in determining emotion conveyed, and such features are not learned by the models. The classification predictions given by the XLM Roberta model may be disagreed with and at the same time agreed by some people, which indicates that the model has learned something regarding the emotion. The perceptual evaluation of the SVM model can help understand the additional information gained by the XLM Roberta model. Typically, music excerpts are annotated for perceived emotion by 15 to 20 annotators to ensure only those experts with high agreement are included in a dataset. This is one limitation of the current study, that the excerpts have been annotated by a limited number of annotators.
Thank you, Dr. Shankar. We have our next presentation by Ms. Catherine Sa and Ms. Pranjali Ektare from KAIS University. The topic is on what catches the eye, priming, bottom-up processing, and visual letter search. Let's move to the recorded video.
Hello everyone, this is Pranjali and Catherine from Christ University. And the title of the poster that we're going to be presenting is Priming, Bottom-up Processing and Visual Letter Search.
Priming is a top-down phenomenon by which exposure to a stimulus facilitates or inhibits the processing of the same or a similar stimulus. Visual search is the process of scanning one's environment to detect a target from among distractors. Previous studies have found perceptual priming effects on visual search. In these studies, exposure to stimuli on previous trials enhanced visual search in subsequent trials. Masked priming effects have also been found in studies of visual letter recognition. In the present study, rather than using inter-trial priming, we used a novel orthographic priming task. We asked participants to take part in the task before taking part in the visual search task. 20 participants were assigned to one of two groups, Group O or Group B. In each group, they were asked to write as many words as they could think of, beginning with their assigned letter in 120 seconds. We expected that this task would activate abstract letter representations. After this, all participants took part in a visual search task. In half of the trials, the target letter was O, and in half of the trials, the target letter was B. This order was randomized across trials. We hypothesized that reaction times for prime letters will be lower than for unprime letters. In each trial, the target stimulus was presented for 800 milliseconds. After this, participants had to detect the target stimulus from an array of distractors. When they had found the stimulus, they had to click the mouse button. Then they had to indicate where on the screen they had seen the target stimulus.
A comparison of mean reaction times across groups shows that the reaction time for B was much faster than for O, irrespective of the group membership. A paired t-test revealed that there were no significant differences in the mean reaction times of two groups. Thus, the direction of difference was not in line with the hypothesis. Also, during the debriefing, participants mentioned that they found it much easier to respond to the letter B in comparison to letter O, as they expressed they confused it with distractor Q.
Participants' experience of visual confusability suggested that the perceptual features of the letters were influencing performance on the task. Additionally, there was an absence of top-down letter frequency effects. Even though the letter O is more common in the English language, participants still responded faster to the letter B. This indicates that in our task, there was a greater involvement of bottom-up processing rather than top-down processing from priming effects and letter frequency effects. One of the reasons for this could be that we used a singleton task. Previous research suggests that singleton tasks may only facilitate short-term priming using bottom-up processes.
By singleton tasks, we mean tasks that only involve looking for a singular feature, as opposed to conjunctive visual tasks, which require looking for an integration of two or more features, such as looking for a letter in a specific color. Though the results clearly defy our top-down expectations, they also aren't completely supported by the bottom-up explanations that exist. Thus, we need to devise experiments that integrate both bottom-up and top-down approaches to help us better understand the complex nature of interactionism between them, which ultimately leads to perception. In the future, we could also employ conjunctive visual search tasks and letters with similar frequencies. As orthographic tasks involve a semantic processing and visual search tasks require visual processing, it would be better to use a neurobiological approach involving a network-driven explanation to help us better understand the phenomenon. In the future, we could look into the validity of the orthographic priming task for a visual search task itself.
In our experiment, as we used 120 trials, and even though they were held constant across all groups, they would have affected data collection. The sample we used consisted only of college students and it was small and unrepresentative. Further, we also had a lack of control over the environmental factors during experimentation, both of which could be rectified in the future. Also, further inquiries could look into the qualitative dimension by taking into account the subjective reports of the participants as they underwent the orthographic priming task. Thank you. The following are our emails if you need to contact us in case of any queries. Thank you for the opportunity.
Thank you, Ms. Pranjali and Ms. Catherine for the presentation. We have our next presentation by Dr. Pooja Sani from Cloud9 Hospital. The presentation is on effect of nature experience on frontoparietal correlates of neurocognitive processes, an ERP study.
Since enhanced attention and awareness was found to be central construct for mindfulness and also sustainable behaviors. We examined attention experimentally by investigating the neural activity during an attentional task. Shown on the bottom of the slide is a typical brain wave. Posner suggested that cognitive subprocesses like detecting a stimulus, inhibiting the distraction present in the stimulus, and eventually selection of a correct choice are elicited in specific brain regions. Challenge here is to design an experiment that evokes these subprocesses and also that we are successful in segregating the corresponding brain responses. Now, another useful model found relevant in context of this research says that involuntary attention is evoked spontaneously. Elements in nature captures our attention without any effort, and this form of attention is argued to involve inner brain regions which are responsible for effective responding. Going back to our hypothesis, along with motivation, the affective state thus induced triggers a series of cognitive processes in the corticolimbic regions associated with learning, selective attention, and cognitive control. In an attempt to map these processes, the state changes after a brief nature experience were observed. Participants were presented with nature stimuli to evoke nature experience akin to being in nature, albeit in a lab setting. The brain activity was examined during the nature experience and while a task was performed. So, findings of this study suggest that nature experience enhances the attention as participants after nature experience were found to overcome distractions more efficiently. As for the subprocesses, there was no significant difference for stimulus detection. However, there was a significant difference found with inhibitory control processing with lesser effort in the after nature experience task. There was also a significant difference for the information processing and resource allocation in decision making. What we also observed was that the overall neural activity is more localized after nature experience, meaning that there were fewer distractions present in the mind and the mind was more focused on the task in hand. Now, this is also explained by enhanced alpha in the frontal and theta in the frontocentral regions of the brain, indicating a state of relaxation and internalized attention. This study is published in Annals of Neurosciences, and to know more details you can refer to this paper. So, altogether evidence is supportive that nature experience is conducive to promoting a conscious awareness and living. These are some of the publications which have resulted out of the research.
Thank you, Dr. Pooja. That was a very interesting presentation. So the last presentation today is by Ms. Vidisha Golecha on designing synthesis and analysis training, impact on innovation abilities in engineering students.
Good afternoon, you all, and welcome. I am Vidisha Golecha, and I completed my engineering integrated course with my major subject as cognitive neuroscience from the Center for Converging Technologies, University of Rajasthan. I am presenting to you my research project from the Center for Creative Cognition, SR University, Warangal, Telangana, under the supervision and guidance of Dr. Raja Shekhar and Sridhar Kundur sir. So, I had like to talk about how design synthesis and analysis training impact on innovation abilities in engineering students. We will discuss the experimental design we used to examine the problem and the results we obtained. Well, we all know what an open-ended question is. And when presented with one, the human brain typically derives solutions from its long and short-term memory. which implies the solution we obtain is a combination of ideas rather than a detailed examination. Thus, instead of analyzing, we simply provide a solution by synthesis. However, this approach is not feasible in fields that require creativity and innovation, such as design thinking. Analysis and synthesis thus form the two fundamental tasks to be done in design thinking. The design thinking process starts with reductionism, where the problem statement is broken down into smaller fragments that can be handled and studied easily. Each fragment is brainstormed and the different smaller solutions are then put together to form a coherent final solution. Literature confirms that designers or engineering students fail to analyze problem statements effectively and start synthesizing them immediately. In this context, the study seeks to reduce fixation and improve creative ideation by combining training, analysis, and synthesis. Hence, our research question is to examine whether students trained in synthesis and analysis show a significant difference in comparison with the data collected before they were trained. In the study, 80 undergraduate students participated from multiple engineering colleges. Before the experiment, the students were subjected to the need for cognition scale to check for motivation and effort to think. Following this, the experiment consisted of two parts involving two groups, each with 40 students. First, a design task, design effective garbage bins, was given to group 1 and the participants were encouraged to propose multiple designs. Then the second group was given an analysis task, that is, list down the pain points using a table fan. Each group had 15 minutes to complete the task. After the task, participants were given 15 minutes of training on analysis, here brainstorming, followed by 30 minutes of training on synthesis, here scamper strategy, which is a design heuristic. Post-training, the task in the first set was switched between the two groups, such that the group which carried out the analysis now carries out the synthesis. Following this, the participant designs were evaluated on fluency, flexibility, and originality. Where fluency is the total number of ideas produced, flexibility is the variety of ideas produced, and originality is the number of new and innovative ideas produced. Subsequently, the participants' designs were also scored for the type of designs generated, whether variant design, developmental design, adaptive design, or original design. 4 distinct designs can be identified based on a combination of conceptual and configurational novelty. The data analysis revealed that the ideation scores, that is flexibility, fluency, and originality scores, were very low in the first session compared to the significantly high scores in the second session, that is following 45 minutes of training. Similarly, the number of pain points listed was very low in the first session compared to the substantially high scores in the second session. Furthermore, the correlation between the need for cognition scale and the ideational metrics, that is fluency, flexibility, and originality, shows a positive trend but is not significant. Also, pre- and post-fluency scores of the male group and the female group show equal ideational capabilities. The following data illustrates the total number of designs collectively from each group before and after training, respectively. Therefore, we observe a steady decline in the number of designs with low conceptual and configurational novelty, while in the remaining domains there is an increasing trend. Thereby, we conclude, as a result of the training, fluency, flexibility, and originality all have shown a significant increase in the synthesis task. As a result of the training, fluency has also been shown to improve in the analysis task. The quality of ideas improved after training, meaning that they became conceptually and configurationally more adaptive and original, and no change in the ideational capabilities based on gender was observed. I thank DST for the financial support for this experiment, Engineering Institutes for permitting to collect the data, the Center for Converging Technologies, University of Rajasthan, and Center for Creative Cognition, SR University, for providing this opportunity. I would like to express my sincere gratitude towards Dr. P.V. Rajashekar sir and Sridhar Kundur sir for their continuous support and guidance during this project. Thank you.
Thank you, Ms. Vidisha. So today's poster session is over. Next, we have a 30 minutes question and answer session. The Zoom link to ask questions to individual presenters are available on the website www.amrita.edu/ACCS8. Under the program schedule, next to the posters titles, please find the links. All the presenters are requested to be online. Once again, thank you all for your wonderful presentation.
Thank you so much, ma'am. That was a very interesting poster session. Each one was very informative and well presented.